Thanks, Dr. Dong, for your introduction, and thanks for having me today. Today, I'm excited to share my work on developing a delivery system called antigen-presenting nanoparticles to deliver mRNA to engineer antigen-specific T cells in vivo. So currently, cancer immunotherapies are transforming, T cell-based immunotherapy are transforming the way cancer patients are treated. So this including immune checkpoint inhibitor, bispecific engager, and engineered T cell therapies. So unlike conventional chemotherapies, usually they provide a non-specific cytotoxicity, and usually the treatment is pretty short-term. But for the T cell-based immunotherapies, they are leveraging our own immune systems, particularly T cells, so they can actually provide more durable treatment response, and also potentially has lower side effects compared to the chemotherapy. And currently, especially the engineered T cell therapy have shown pretty promising, even curative treatment outcomes in certain type of cancers. So a key player behind these T cell based immunotherapies are antigen-specific T cells. So, here we are going to use viral infection as an example, but the same principles are applicable to T cells for other diseases such as cancer and autoimmune disease. So when we have viral infection, those viral antigen-specific T cells, they recognize infected cells by using their T cell receptor, which we call TCR. And so the TCR will recognize the viral peptides present by the MHC, a protein expressed on the cell surface. So this recognition would activate the T cells and then trigger them to release cytotoxic molecules such as paraffin and granzymes to kill the infected cells. So this will facilitate the clearance of viral infections. So once the viral infection is clear, a small portion of antigen-specific T cells will become memory T cells. So this population will protect us from the next time when we encounter the same type of infection. So simply put, antigen-specific cytotoxicity provided by these T cells help us clear diseases and also provide a foundation for T cell-based immunotherapy. And the memory T cells protect us, provide more long-term immune protection. So the vaccine development focus on generating antigen-specific memory T cells. So in our body, there are actually a lot of different T cell population with their receptor recognize antigens associated with different disease settings, such as cancer, viral infection, and autoimmune disease. And even in a given disease setting, there is also a collection of different antigen-specific T cells with their receptor recognize different peptide antigens, such as in a common viral infection, EBV. The EBV-specific T cells, their receptors can recognize more than 40 different peptide antigens. So, in our body is actually a lot of millions of different antigen-specific T cells. But the current cancer immunotherapy, they don't only target cancer-specific T cells. For example, the immune checkpoint blockade therapy, which shows can cure about 1/3 of patients with myeloma and others cancers. But the problem is those checkpoint inhibitor therapies, such as anti-PD-1, they would activate T cells express PD-1 surface protein, regardless the antigen specificity of their T cell receptor. So they broadly activate T cells. These would lead to the problems, including autoimmune reactions and sometimes dose-limiting toxicity. So these adverse effects is not just encountered by the immune checkpoint inhibitors, but also cytokines and bispecific T cell engager, these immunotherapies. So to avoid those adverse effects, here we ask whether we can specifically, selectively only target, preferentially target and activate cancer antigen-specific T cell in vivo, such that we can avoid the adverse effects. So to answer these questions, we have been working on a delivery system called antigen-presenting nanoparticles, or APNs. So we learn from the endogenous interaction between the peptide MHC and T cell receptor between the infected cells and virus-specific T cells. So what we did is we express recombinant peptide MHC and then functionalize on the surface of lipid mRNA-loaded lipid nanoparticles. So just as a side note, this lipid nanoparticles is pretty similar as what we get the COVID vaccine, the Moderna or Pfizer COVID vaccine. So it's very clinical relevant. So we envision that when we inject APN to mice and potentially in the future for patients, those APNs would selectively target and deliver mRNA to their cognate antigen-specific T cells and to program their activity, while minimize the mRNA delivery to non-cognate off-target cell populations. So this ability to deliver mRNA to the selective antigen-specific T cells will allow us to begin program T cell activity by delivering mRNA encoding such as cytokines to enhance their effector function to kill cancer or knock down the immune checkpoint PD-1 expression, which would inhibit the T cell activity. So we can avoid this from happening. So, so to study whether our APN can target and transfect antigen-specific T cells, we first start from 3 models, mouse models, which is called T cell receptor transgenic mouse model. So in those mouse model, their CD8 T cells have only one kind of T cells recognize the same peptide epitopes. For example, in the P14 mice, all the CD8 T cells recognize these GP33 peptides. And female transgenic model recognize human myeloma GP100 peptide. In the case of OT1, they recognize OVA peptides. So using those TCR transgenic mice model will allow us to see whether APN can selectively transfect antigen-specific T cells. So we first look into whether APN can target antigen-specific T cell in the ex vivo setting, which means we take cells out of the mice and then do the experiments outside the mice. So here we use P14 TCR transgenic mice. To do these, to study these, we make 2 different types of APN, one with GP100. So these would be a non-cognate or non-targeting control. And the APN with GP33 peptides, which is a cognate APN, which theoretically has the ability to target P14 CD8 T cells. So we label the APN with fluorophore, so we can track whether their interaction with T cells by flow cytometry. So we can see after co-incubate with T cells, the T cell treated with PBS or non-cognate GP100 APN, they don't really show any signals from the APN. In contrast, more than 95% of the P14 CD8 T cells shows the signals from GP33 APN, which is significantly higher than the non-targeting GP100 APN, suggesting the GP33 APN in this shows the targeting to the P14 CD8 T cells. We also use POT1 transgenic mice model to show these antigen-specific targeting in the ex vivo setting. So next, we want to see whether these APN and the CD8 T cell interaction actually induce the particle uptake by the T cells. So we did an acid wash study where we apply acid to strip to remove any APNs binding to a surface, but not in the cells. So we did this study at 4 degrees C and 37 degrees C. Because the T cell receptor mediated uptake, internalization is ATP dependent. So we would expect higher internalization at 37 degrees C. So using flow cytometry, we can see after acid treatment, the particle signals in the T cells reduce after acid wash at 4 degrees C. But 37 degrees C, with and without acid wash, we don't really see a significant difference. So suggesting that the APN indeed internalized by the particles, otherwise we would see a reduction of the particle signals in the T cells. So after confirming that, next we want to see whether APNs can show transfection inside the mice. So what we did is we encapsulate APN with mRNA encoding firefly luciferase, which can convert substrate into bioluminescence. So we take our organs and then use IVs to detect the bioluminescence from the major organs. So we can see compared to non-cognate APN, cognate APN shows the significant higher signals in the spleen compared to other major organs. So just for people who are not familiar with immunology, spleen is the second largest immune organs. There are 20% of the sphenocytes, 20% of the spleen cells are the CD8 T cells, which can be targeted and transfected by our APNs. So we hypothesize this is because there are a dense of high number of target cells that can be transfected by APN in the spleen. So that's why we can see the transfection localized in the spleen. So next, we ask whether APN can transfect CD8 T cells in mice. So similarly, we use P14 TCR transgenic mice model. And instead of using firefly luciferase, we use VHH mRNA. So VHH we use as a model protein. So we can use antibody to stand the VHH expression on the cell surface as a way to know the transfection efficiency in the CD8 T cells. So similarly, we label APN with DAID fluorophore to track the targeting of APN to the CD8 T cells. So we can see compared to non-cognate, non-targeting control, the cognate APN shows targeting to more than 97% of the CD8 T cells in the spleen and achieve more than 90% transfection in the T cells in the spleen. Notably, in the CD8 negative, which are the off-target cell population, we don't really see any transfection of target transfection. So which suggesting the specificity of our APN to transfect CD8 T cells. But to really break down to see the, because in the P14 transgenic mice model, is actually more than, is about 20% of the sphenocytes all the CD8 T cells are showing the same T cell receptor. But in the physiological condition, the antigen-specific T cell frequency is not as high. Usually it's about 1 to 5%, depends on disease setting. So to better mimic the physiological condition, we use PR8 human influenza mouse model. So we make two different kinds of APN. So it's APN either carrying MP366 or PA224 peptides to target the top two most immunodominant flu-specific T cells, MP366 or PA224 CD8 T cells in this PR8 influenza mouse model. So we can see we dose the PR8 infected mice with two different mRNA dose. We can clearly see a dose-dependent targeting and transfection in the top two immunodominant flu-specific T cells. So notably in these MP366 negative and PA224 negative T cell populations actually involve millions of different T cell clones. So the fact that we can selectively target and transfect the flu-specific T cells in the midst of other millions of different T cell clones is actually very powerful to allow us really start looking at how can we selectively transfect the disease-relevant immune CD8 T cells. So next, we want to see how precise the APN transfect antigen-specific T cells among other off-target cell population. So we look at spleen and liver, which are the two major organs nanoparticle distribute to. So again, we use PR8 influenza mouse model, and then use PA224 APN to target PA224 flu-specific CD8 T cells. So as expected, APN shows some transfection in macrophage, monocyte, and Kupffer cells, because those cells tend to uptake any foreign materials from outside of the body. But what makes us excited is that APNs achieve significantly higher transfection in these PA224 specific CD8 T cells. And again, in the other millions of different T cell, CD8 T cell population, we didn't really see any off-target transfection, suggesting the APN can selectively transfect antigen-specific T cells. As I mentioned earlier, in our body, there are millions of different antigen-specific T cells, and in a given disease setting, there are also a collection of different antigen-specific T cells. So in order to better program cancer-specific T cells, we will need to be able to program as many T cell population as possible. So we would expect we would need to target like millions or even hundreds of different antigen-specific T cells. To do this, we will need the MHC presenting different peptide epitopes. If we are targeting 20 different antigen-specific T cells, we will need 20 different peptide MHC. So to make one kind of peptide MHC, it takes about 1 week from protein expression, refolding, and purification. So imagine we need to make tens or even hundreds of different peptide MHC would be very labor intensive. So we look into a protocol which is called UV-mediated ligand exchange, in which we just need to make a big batch of peptide MHC with UV labile peptide. So we can simply shine an UV light to cleave the peptide, the dummy peptide, in the presence of other peptide epitopes. So this will allow us to quickly swap the dummy UV peptide into different peptide of interest in less than 1 hour. So we incorporate this efficient exchange protocol into our APN synthesis pipeline. So we conjugate lipid tail to this UV cleavable peptide MHC, and then swap the UV peptide into different peptide of interest, and then do a post-insertion to modify this peptide MHC on the lipid nanoparticle surface to make this APN library. So the beauty of this approach is that we can avoid the mRNA degradation by the UV exposure, and also with this APN library, we can achieve a multiplex mRNA delivery to multiple clones of antigen-specific T cells. So we first want to make sure that UV exchange APN shows comparable targeting and transfection compared to the folded APN, like the data I showed you earlier. So we first using the 3 TCR transgenic mice model, and then we can see compared to the conventionally folded APNs, the UV exchange APN shows comparable targeting across these 3 TCR transgenic mice model. And using the PR8 flu infection mice model, as I mentioned earlier, we can also see comparable targeting and transfection across these between these folded and UV exchange APNs. So this data suggests using UV exchange APN, we still can achieve a comparable targeting and transfection compared to the conventionally folded APN. So the next question is whether we can achieve the mRNA delivery to multiple antigen-specific population simultaneously. So to test that, we use the UV exchange protocol to make a 3-plex APN library with MP366, PA224, or GP33 that can target the top 3 immunodominant T cell population in these PR8 GP33 flu infection mice model. So just of note, the frequency of these 3 antigen-specific T cells ranging from 1 to about 4% of total CD8 T cells in the spleen. So even in this such low frequency, APNs were able to transfect the top 3 immunodominant flu-specific T cells with comparable transfection efficiency. And again, we don't see detectable transfection in the rest of millions of different T cell populations. So in my talk, I show you the APNs were able to preferentially transfect T cells in the spleen, which is the second largest immune organs, and also the APNs were able to achieve antigen-specific T cell transfection, and this delivery specificity has now been achieved by other targeting approach, including antibody and chemistry-based targeting. And we were able to achieve mRNA delivery to multiple 3-plex flu-specific antigen-specific T cell subsets. So with this tool in hand, now we are looking at using APN to improve cancer treatments and against autoimmune disease, such as type 1 diabetes. So with that, I would like to thank my advisor, Dr. Gabe Kwong, for his advice, and also previous and current lab members in the Kwong lab, and also our collaborator, Dr. Phil Santangelo, and his lab members. And also thanks Dr. Rafi Eman for his advice on the T cell biology and the T cell models. So we are currently working with Dr. Madhav Dotekar at Winship Cancer Institute to use our APN to improve treatment for the multiple myeloma. Also, thanks my funding agents for their support and the excitement about our projects. Thank you. I will be happy to answer questions.